Okay, so let's get started. Hello, everyone. Welcome to our today's Immune seminar. So it's absolutely my pleasure to welcome our today's speaker, Dr. Rick Maizels from UK. So Rick currently is full professor of parasitology in University of Glasgow. So a very brief introduction for Rick. Rick complete his undergraduate study in zoology in University College London and continue with PhD training in immunology in National Institute for Medical Research, also in London. After which, Rick pursued his postdoc training in microbiology in UCLA. So since that, Rick moved back to UK, and then 2016, Rick actually become a full professor of parasitology in University of Glasgow, and then almost as similar time, 2022, Rick also appointed as director of research, School of Infection and Immunity in University of Glasgow. So Rick's lab is really did a substantial amount of work in parasitology. And then his lab is really mainly aimed to understand the molecular basis of how parasites evade the mammalian immune system. His lab seek to integrate detailed studies at the level of individual parasite molecules with system immunology, which measures responses not only against the parasite, but also against the bystander antigens such as allergens. His lab developed a lot of new technology techniques, combined DNA analysis, gene isolation, protein expression, and function studies, and immune system biology to really comprehensively understand how the interaction between the parasite and then the host. More in particular, his lab currently testing the hypothesis that Helminth parasites exploit the body's own safety machinery, which have involved in minimize the risk of autoimmunity, such as how the regulatory cells naturally arise to limited autoreactivity, but also associated with chronic Helminth infection. So, as I said, Rick's lab actually really contributed significantly in parasites immunology for many groundbreaking discoveries. Rick had numerous awards, which including Eurolife medals. He was elected as Fellow of Academy of Medical Sciences in 2017, and then also Lukart Medal of German Society of Parasitology on 2020s. So I think without further ado, it's really a pleasure to have you on our platform, Rick. Please go ahead.
Oh, thank you so much. Thanks. Very kind invitation. And it's a real delight and honor to be invited to give this seminar. So thank you for that. So I'm going to talk to you about Helminth parasites, which are the multicellular worms that are able to invade our body and survive for a long time in the face of the immune system. And one place to start is considering the global distribution of different diseases. And there's a reciprocal relationship between the areas which have high levels of parasite infection. My pointer's not working on the left. And those other parts of the world where more inflammatory diseases, autoimmune diseases and allergies, here we have asthma on the right-hand side, are much more prevalent. And so a big question in the field has been whether these are in any way, pardon me, causally related. In other words, there are so many environmental factors that are at play in different parts of the world. Are these inflammatory diseases suppressed by helminths or are they enhanced by many other factors? So one of the key developments in understanding this came from Maria Yazdanbash's lab in Leiden, where they tested children in West Africa, and children with a schistosome helminth infection on the left here in blue had a significantly lower level of allergic reactivity than uninfected children. So this began to spark off the idea that perhaps Helminth parasites, through the bystander effect that was just mentioned, could have a systemic suppressive effect on inflammation. And then following Maria Yazdan-Bash's inspiration, in our lab, we tested in mouse models whether a parasite, and I'll come to the parasite in particular in a moment, whether a Helminth parasite could suppress airway allergy. And on the right, you can see here with the mouse model that mice sensitized and challenged with DERP1, the house dust mite allergen, that's DD. Those mice showed a very high eosinophilia in the airways, an allergic eosinophilia. But if they were carrying an H. polygyrus Helminth infection, they were protected. And we were not the only labs looking into this. There's been a really wide range of studies in many different systems showing that it's a really robust effect that the presence of a Helminth parasite dampens, to some degree or another, the inflammatory and allergic and autoimmune reactivity of the host. And I can sort of boil this down very, very simplistically to this model here, where we attribute much, but not all, much of the dampening of inflammation to the activation of regulatory T cells. And this begins to explain why the Helminth infections seem to be able to suppress both type 2 disorders, such as allergy and asthma, and type 1 disorders, such as colitis and MS. It's not the whole story. And at the end of the presentation, I'm also going to show you a little bit of data which implies that parasites also can modulate their environment irrespective of T cells by directly acting on the intestinal epithelium. I think next I should tell you about this parasite H. polygyrus. And this is, to us, a very, very useful model system. It's a natural parasite of mice, and so it's well adapted. It understands the host immune system very well. It's very adept at manipulating host immunity. And it has a life cycle where it enters orally, so number one on the left-hand side, into the lumen of the small intestine, burrows through the epithelium and forms a cyst, and the host forms a granuloma in the submucosa, and the larva grows there for a week, and then at day 8 or 9, it exits as a fully grown adult. So on the right-hand side, you can see a cross-section of one of these granulomas with the tightly coiled worm in a section. And that's something I'll return to later in the talk, this granuloma. And then the final right-hand image is of the adult worm in the lumen of the gut, and you can see the villi. So to give you a sense of scale, these are parasites that are macroscopic. You can see them with the naked eye. They are very large. And one of the points here is that the parasite is breaching the epithelial barrier twice at day 0 to 1, and then again from day 8 to 9. So this is a major challenge to the immune system. So what does the immune system do? Well, it generates a Th2 response. As most people will be aware, Helminths and Th2 go hand in hand. And the level of the Th2 response is a factor in whether mice are resistant or susceptible. So here you can see CBA and B6 mice are very susceptible, but SJL mice are very resistant, and BALB/c are somewhat on the cusp. They hold the infection for a few weeks and then they can expel it. And then the granulomas that form in the wall of the gut appear to protect the host against infection, because the resistant strains form larger and more potent granulomas. And they're really striking in this image I've got here. And these are essentially how the host can trap the parasites and prevent them from returning into the lumen. This is the Th2 response, but there's also a regulatory T response, and the Treg response for example, in the B6 mouse is responsible for its susceptibility. So we showed that if you deplete Tregs in B6 mice, you make them resistant. And then bulb C mice, we could make more susceptible by giving them IL-2 complex, which is a sort of short way of expanding the regulatory T cell compartment. And when that's done, in the bottom left here, you can see bulb C plus the IL-2 complex show a much higher worm burden at day 28, when normally they've been able to clear most of the worms. And at the same time, those mice that have expanded their Tregs show far fewer of these granulomas. So there's a dynamic in which the Treg expansion favors parasite survival. And that made us ask the question, well, do the parasites induce or expand Tregs as part of their survival strategy? And we believe that is the case. So let me just summarize. If you ask what happens to the Treg population in an H. polygyrus infection, it increases stepwise. So you can see here it goes from roughly 12% of mesenteric lymph node cells to around 18%. So maybe that's not a dramatic change, but you have to think that it's the ratio of regulatory T cells to effector T cells that determines the outcome. So from 1 Treg to 7 effector cells, you're changing from 1 Treg to 5 effector cells. That may make the difference when the infection is on the cusp of survival or expulsion. But more importantly, we asked the question whether this expansion of FOXP3 cells was due to the presence of the parasite. And indirectly, we decided to approach that by examining what we call ES products. And this means excreted stroke secreted. In other words, products released by parasites into their environment. And this is a concept which says that because the parasite population is essentially continuous, it's established that its relationship with the host must be through released products from the live worms. So we took live worms and simply put them in serum-free medium, collected the conditioned medium, and asked whether there was a substance in the conditioned medium that expanded Tregs. And that was the case. So in this graph, what we did is we took the secreted materials, ES from H. polygyrus or HES, and added those to mouse T cells. And the T cells have been sorted, so they were FOXP3 negative. And in the bottom, you can see that after 3 days, 50% of the T cells had switched on FOXP3. And the control we use is TGF-beta, which is the known inducer of FOXP3 in T cells. And you can see that essentially the HES phenocopies TGF-beta. And other work which we published said that if you conducted this experiment in the presence of the inhibitor of TGF-beta signaling, you lost FOXP3 induction by the parasite. So the parasite was using the TGF-beta pathway. So next, we took this conditioned medium, which is obviously a complex of many, many different molecules, and we screened it with a cell line that reports TGF-beta receptor ligation. So this is a schematic of this cell line where TGF-beta, a dimer, binds to TGF-beta receptor 2. That's a kinase that phosphorylates receptor 1, another kinase which activates the SMAD signaling pathway. And the cell line has an alkaline phosphatase reporter of SMAD binding. That means you can measure the presence of TGF-beta ligation essentially on an ELISA plate looking for the enzyme. We did this, we found a parasite protein that activates this cell line, and we called it TGM. You'll see why in a moment. And the TGM in green is active and much more active than TGF-beta in the sense of it drives a higher level of alkaline phosphatase released from these cells. So why is it called TGM? It is because it's a mimic, not a homolog. So a homolog would be evolutionarily related with sequence conservation, but the parasite product is completely different. And so the TGM I've shown here has 5 domains. I'll come to these domains in a moment. TGF-beta on the left here is a homodimer, I just mentioned that, and it binds receptor 2, and then that combination binds receptor 1. So there's no sequence similarity between mammalian TGF-beta and the H-polygyrus TGF-beta mimics. Moreover, the TGF-beta mimic, TGM, has a modular design in which these 5 different domains act as independent moieties that can be added or removed from the protein, and each domain carries with it a specific activity. So in this case, TGM1, domains 1 and 2 bind TGF-beta receptor 1, and domain 3 binds TGF-beta receptor 2. And we had a fantastic collaboration with a group in Pittsburgh, Andy Hink's lab. And his lab, I'll show you some more data in a moment, but his lab also characterized biophysically the binding of these domains to receptor 1 and 2, showing without doubt that there's an intimate molecular interlocking of these domains with the mammalian receptors, and characterizing the on rate, off rate, and the affinity of the parasite protein for the mammalian receptor. So further work that Andy Hink did in Pittsburgh shows this mimicry at a molecular level, at almost an atomic level. So here, what we have is a comparison of how the mammalian TGF-beta binds to the TGF-beta receptor, and compare that to the parasite mimic. So the blue is the same receptor, mammalian receptor. The pink is TGF-beta, mammalian, and the green is TGM, parasite. So you can see, if you look at how the beta sheets are organized, it's completely different architecture between the TGF-beta and the TGM. But then if you look at detail at the interface, the TGF-beta and TGM are closely related. So you have at the top a salt bridge between the aspartic acid and the arginine, and beneath you have another bridge between glutamic acid. And then the binding also requires a hydrophobic pocket around the isoleucine 53 of the receptor, and both TGF-beta and TGM can accommodate that. So it's an astonishing convergent evolution in which the parasite has formed a protein that can mimic the face of TGF-beta while retaining a completely different support structure behind. So this protein from the parasite is a very effective mimic of the function of the protein too. So here are some examples. I've shown you the effect on the reporter cells on the left already. You can take the TGM1 and it activates CD4 T cells from mouse or from human to induce FOXP3. And we can make Tregs in the lab using the parasite protein, transfer them into mice, and it protects them against EAE. So it's certainly as effective as TGF-beta, and I'll show you in a minute some features which take it, we think, beyond TGF-beta. But it also can act as a direct anti-inflammatory in vivo. So here's an example where we use a animal model of airway allergy. So very similar to the experiment we did at the very beginning, where we took mice and infected them with live parasites. In this case, instead of infecting them, we are giving them the parasite protein, TGM, during the challenge when the immune response, the allergic immune response is being mounted. And when that happens, the eosinophilia is essentially abolished due to the presence of the parasite protein. A different type of experiment would be intestinal inflammation, a model of IBD, in which transfer of T cells to a rag mouse slowly develops into an inflammatory disease due to the reaction against commensal bacteria in the gut. And in this case, we gave the TGM protein at the onset of symptoms when mice began to show inflammatory parameters. And when they're given TGM in here, and you can see where the gray shading is, they were significantly protected. However, when we stopped giving them TGM, they caught up and became similar to the untreated mice. So in this case, the TGM alleviated the disease for a window during it was present, rather than gave a lasting effect. Nevertheless, we can show the same protein acts against a type 2 response in allergy and a type 1/17 response in colitis. That's sort of background that TGM is a functional mimic protein. And now I need to add another layer of complexity. And that is that you may have noticed in some of the slides, it actually says TGM1, not just TGM. And the reason is there are multiple proteins from the parasite of this that are similar to TGM. So there are 10. And so this was always a bit of a paradox. Or why wasn't the parasite satisfied with the TGM I've just been telling you about? It seems to work pretty well. Why would it need additional TGMs? And we also were looking in terms of these 5 domains, that some proteins had lost those domains. So if you look, TGM 5 and 6, 9 and 10 have lost some domains, and others have gained them. So there seems to be a flexibility in the domain organization of this protein. But the other feature to draw your attention to is the variability in amino acid sequence. And so in each box, the figure relates to amino acid identity relative to TGM1. So if you scan down domains 4 and 5, so the beige and the magenta colors, the identity really becomes very divergent. So there's only 30% amino acid identity with TGM1. So I'll come back to that shortly. So our first foray into asking about this was to investigate the role of domains 4 and 5, because TGFB receptors 1 and 2 only bind to domains 1, 2, 3. And we found that domains 4 and 5 bind to another receptor, and you can see it there, CD44. So this started when we were trying to truncate the TGM protein down to its bare minimum that was necessary for immune modulation. And we found, curiously, that if we removed domains 4 and 5, the protein was still active, but it was 10-fold less potent. So we shifted the curve to the right. And then we started collaborating with another fantastic group in Leiden in the Netherlands, Peter ten Dijk, and Martin van Dinther in his lab, used domains 4 and 5 alone and did pull downs to identify if any other protein from mammalian cells interacted with these domains. And that pull down, subject to mass spec, found one protein, is here with the blue arrow, CD44. And then a postdoc in our lab, Shashi Singh, repeated these experiments and showed here that you could pull down with full length, that's domains 1, 2, 3, 4, 5, TBR1, TGF-beta receptor 1, TGF-beta receptor 2, and CD44. But if you split the protein into domains 1, 2, 3 and domains 4 and 5, domains 1, 2, 3 pull down the TGF-beta receptors, and domains 4 and 5 pull down CD44. So it was a separation, and we had a function now for domains 4 and 5. Then it took us back to our conceptual model that, well, domains 1, 2, 3 bound to the canonical receptors and triggered the SMAD signaling pathway I've talked about. Domains 4 and 5 bound to co-receptors. And going back to the idea that the amino acids, its conservation in 4 and 5 is relatively low, I will show you that domains 4 and 5 encode different receptor specificities for the different members of that family. So this allows the TGM family to mediate cell-specific TGF-beta signaling. So here's an example. One of the additional TGMs is called TGM4. And this was curious because it did not activate the fibroblast reporter cell line that we had depended on. So this was found by it's a nucleotide and amino acid sequence similarity, not by screening for function. But when we made it, it still didn't work on fibroblasts. And in the center, you can see pSMAD, Western blot. There's a tiny amount of activity with TGM4, but way down on TGF-beta and TGM. And the right-hand bar graph is just 3 replicate experiments densitometry, showing there's a very significant loss. But so rather than throw TGM4 away, turns out actually it will induce FOXP3 in T cells. So it's better on T cells than on fibroblasts. And it's very good on myeloid cells. So myeloid cells, if you look at the bottom central panel, pSMAD Western, it's just as good as TGF-beta and TGM. So somehow this protein is selective for immune cells and particularly for myeloid cells. And so Andy Hink's lab took another look at TGM4 and measured the affinity for the TGF-beta receptors. And if you look on the very right-hand side, affinity for TBR2 in red, the remarkable finding was that the TGM4 is 30-fold less strong affinity for the TGF-beta receptor 2. And this puzzled us because why would a parasite evolve a weaker ligand? And so our rationalization of this is that actually by having a weaker affinity for the TGF-beta receptor, it becomes more dependent on co-receptors. And so its specificity is contingent on having a relatively low affinity for the TGF-beta receptor. So I can return to this, but it's more nuanced. In other words, the parasite hasn't necessarily evolved to maximize every interaction with every parasite, with every host target. It's measured it, calibrated it so that it functionally operates in the most selective manner. TGM4, but we also looked at TGM6 and 7. So TGM6, I'm going to say a little bit more about because it's lost these TGF-beta receptor 1 domains. And when we do pull downs, indeed, you can see the TGF-beta receptor 1 Western blot is blank, but TGF-beta receptor 2 binds strongly if domain 3 is present, not if domain 3 is missing. And then the really surprising thing is it binds a completely different co-receptor, LRP1, that's lipoprotein-like receptor binding protein 1, and that's domains 4 and 5. And then going back to the boxes, you can see that domains 4 and 5 are only 30 to 40% identical to TGM1. So they've diverged and evolved a completely different binding specificity. Why? I'll show you in a minute. And just very quickly, TGM7 has a third binding activity. It binds receptors, TGF-beta receptor 1 and 2, and GP130. So that's the IL-6 signaling transducer. It's also shared GP130 with like IL-11, oncostatin, and others. So let me just go to TGM6. It lacks TGF-beta receptor 1 binding. So we thought, well, that's strange. Maybe it doesn't need receptor 1 to activate signaling. We couldn't activate the SMAD signaling in any way with TGM6. Actually, it turns out it's an inhibitor. So on the first graph with the green title, you can see that you can see that TGM1 in blue or TGF-beta in green will activate the fibroblast. This is the reporter cell line. But as you titrate in TGM6, the signal declines. And so before long, the activation of this cell line is abolished by the presence of TGM6. It's an inhibitor, but it only works on fibroblasts. It doesn't work on T cells. And so the central graph here, TGM1 is and TGF-beta are pretty much flatlining. You don't see an inhibition. That relates to the fact that fibroblasts have low levels of CD44 and high levels of LRP1, and T cells have high levels of CD44 and don't express LRP1. So this means the parasite has a TGF-beta inhibitor that is cell-specific, which hasn't been discovered before. The right-hand graph is simply if you take domain 3 alone of TGM6, so just the domain that binds TBR2, it's non-inhibitory. So that the inhibition requires the co-receptor binding. So whether that's an avidity thing or some sort of sequestering of the TBR2, we don't know, but you definitely need the entire suite of domains for the inhibition. And just for completeness, we also knocked out LRP1 in the fibroblasts, and the inhibition is abolished. And this is a model that Andy Hink did for AlphaFold, showing that the TGM6 domains 4 and 5 fit very neatly into the structure of LRP1. So I have a story that the TGM6 is LRP1 specific and can't inhibit cells through CD44. So Peter ten Dijk in Leiden did a really neat experiment, and I borrowed this from him. And that is if you replace domains 3 and 4, sorry, the yellow and the orange here should say domain 4 and domain 5. If you replace TGM6 domains 4 and 5 with TGM1 domains 4 and 5, you change it from becoming a LRP1 dependent inhibitor. So on the bottom left, you lose inhibition in a knockout of LRP1, and it becomes a CD44 dependent inhibitor. On the bottom right, the inhibition is dependent on is lost in CD44 knockouts. So this validates the idea that we've got a modular system that we can chop and change and exchange different domains and rewrite the specificity of agonists and antagonists. So to try and summarize this part of the talk, I've got a little bit more, but this is the main part of the talk. We're saying conceptually that the parasite has evolved a new way of interacting with cytokine receptors through combinatorial engagement. So instead of singling out a single receptor for activation, it uses combinations. And that allows it to change its biological function. So TGF-beta itself mediates many, many different activities on many different cell types. Generally, it's good for immune tolerance, tissue repair, and I'll come to some negatives in a moment. TGM4 is a myeloid preferential signaling protein. We've also got other data I'm not showing you that it interacts with integrin and CD206, but through a combination of these receptors, it interacts with immune cells, but would not activate fibroblasts. And the 6, one I've just talked about, is an inhibitor which inhibits fibroblasts but doesn't interfere with macrophages and T cells. And the bottom line of this is that evolutionarily, the parasite has calibrated the affinity of each of these interactions just to get it to the sweet spot that only some cell types will respond to the ligand. And so just to wrap up this part of the talk, TGF-beta is a very paradoxical cytokine because although it's beneficial in dampening inflammation and establishing tolerance, when it's overexpressed in cancer or in tissues, it can be very detrimental. And so therapies aimed at the TGF-beta pathway are very problematic because they target both beneficial and harmful pathways. And for example, using a kinase inhibitor to accompany immunotherapy results in damage to heart muscle. So only these parasite mimics offer some form of cell specificity that might allow us to separate benefit from harm. So I've got a few more slides, but this is I think this area of mimicry and manipulation and selectivity is really something that is very exciting for us in our lab and our collaborative labs, so that we're trying to not just explore, but transform into potential therapies. So let me just very quickly cover a couple of other points, and I'll try and finish in a few minutes. So we know that immunity, and I showed you at the beginning that Th2 is important and Tregs counter it. Immunity to these parasites requires both type 2 cells, type 2 cytokines, but also the recruitment of the intestinal epithelium as an effector tissue. And part of our epithelium are specialized cells, and those specialized cells would include tough cells, which are both early detectors of the presence of parasites, releasing IL-25, activating ILCs, releasing IL-13 that expands tough cells. And the tough cells also release small pharmacological mediators that activate the immune system and inhibit parasite growth. And if you don't have tough cells, you fail to expel intestinal parasites. And this graph is actually a different parasite from the one I've talked about. This is a rat maintained parasite that's normally expelled. So in the red lines, you can see that it survives only a week in mice before expulsion. But in the absence of tough cells, the parasites endure for 6 weeks or more. So what happens in the H. polygyrus infection I've been talking about? Well, actually, the H. polygyrus parasite isn't subject to such a strong tough cell response. So the left is just some stained sections in which tough cells are more numerous in N. brasiliensis than in H. polygyrus. On the right, we've co-infected H. polygyrus mice with Nipostrongylus brasiliensis. And if you can see that the blue circles is the response to Nipostrongylus, yellow is the response to H. polygyrus, and the green is the co-infection. So the presence of H. polygyrus drags down the tough cell response, so suppresses tough cell responses. It's not that the H. polygyrus fails to stimulate, but it actually interferes with tough cells. And we can do this in vitro organoids, in which taking small intestinal stem cells which self-organize into mini guts or organoids, and in the presence of IL-4/13, you see a vast expansion in DCLK1 tough cells. But if you add the ES from H. polygyrus, the secreted products, that's suppressed. So you can see in the third bar, HES is very few DCLK1 positives. And doing a mRNA survey, we found that the tough cell genes like Pu2F3, which is the transcription factor knocked out in the mouse experiments, and others are all downregulated in the presence of Hess. So the parasite downregulates the tough cell lineage. And actually other lineages, I won't go through this in detail, but also Paneth cells. And we know this because if you look in sections of the granuloma, which I showed you at the beginning, the locality of the granuloma containing the parasites lacks Paneth cells. And here Paneth cells are being stained with anti-lysozyme, and you can see some in the crypts to the left, but the crypts close to the parasite itself have lost their Paneth cells. So in summary, this is showing that the parasite is able to interfere with the fate of the intestinal cells. So intestinal stem cells, instead of becoming Paneth cells, goblet cells, and tough cells, become enterocytes. A way we look upon this is twofold. So one is that the parasite is eliminating the functional epithelial cells that can expel the worms. So goblet cells are making mucus, Paneth cells are making antimicrobials, tough cells are making acetylcholine. And so the parasite is minimizing damage to itself by diverting differentiation towards enterocytes. But the other really interesting concept is that the enterocytes proliferate and are responsible for repairing the breaches that I mentioned at the very beginning, where the parasite penetrates the wall of the gut both at day 1 and day 8 in its entry and exit from the submucosa. So perhaps the parasite has evolved a way at repairing the damage. And another way of visualizing this is the morphology of these organoids. So normally these organoids are budding, and that you can see the bottom left image. They're budding, and each bud represents a mini crypt in which the specialized secretory cells are forming. But in the presence of parasite material, you get these spheroids, which are bubbles, balloons, little structure, and they are the proliferating enterocytes, which form a continuum without differentiation. Strikingly, this image I've got here, the bottom right-hand image, mirrors a publication from a group in UCSF, Ophir Klein and Richard Loxley, in which they took stem cells from an H. polygyrus infected mouse and equally saw these proliferative spheroid organoids, again arguing that the parasite's inducing a repair-like or fetal-like phenotype. Okay, I think I'm running out of time. I don't need to show you this. This is preliminary. I just say that what I've been talking about really relates to a single pathway that the parasite has found to manipulate the host. It is maybe just one small part of the whole story. And if we look at this spectrum I've got here of the different steps and stages of the immune response, and all I've really talked about is the induction of Tregs by this one protein, HPTGM. And there are so many other things going on in a complex infection. Sure, we could look at many of these other mediators, and many labs are looking at these other mediators, but I think equally an in-depth dive into the molecular interactions is so fascinating that we're finding it rewarding just looking at the TGM itself. But if people are interested in any of these others, I can answer. So just to say, these are members of the lab. It's been a really great collaborative team. And then we've done a lot of work with the group in Pittsburgh, you can see here, led by Andy Hink. The team in Leiden also, Martin van Diensten, Peter ten Dijk, and many of the others I've put on this slide. So thanks. I hope I've left some time for questions. I'll be very happy to answer if there are some.